thank you for joining us and welcome to our Research Connect series. It's time to get started. My name is Aisha. I'm a CDTRP trainee and I'll be facilitating today's webinar. Before we get started, first I would like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwa, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant community. I encourage you to write your land acknowledgement in the chat box. We would also like to acknowledge AstraZeneca and CIHR, whose generous support we could not have had to host these sessions without. And before we dive into the main content today, I will pass it to Erica for some general CDTRP inquiries. Updates.
Thank you, Aisha. Today I have two announcements for you all. Our Indigenous platform is hosting a Café Scientifique webinar called Pathways to Truth and Reconciliation in Organ Donation and Transplantation. This event will be held on February 27th from 12 p.m. to 4 p.m. Eastern Time. Dr. Tate will focus on Indigenous people within Canada, and then again from 6 p.m. to 8 p.m. she will focus on Indigenous people outside of Canada, specifically Australia and New Zealand. This event is free, and I will provide the link in the registration link in the chat box below. The second announcement is to save the date for our PFD Forum. This year's Patient, Family, and Donor Research Forum will be held virtually on June 13th and 14th. Registration will be free and can be done through our website. The link to register will be live soon, so keep your eyes out for that. Thank you.
Now, before I introduce the speakers, I have a few housekeeping notes for everyone. Today's session is recorded and posted on the CDTRP website and YouTube channel. During the presentation, please keep your mics off. If you have any questions or comments, you can raise your hand with the raise your hand button or type it in the comment section. Today, I'm pleased to introduce our presenter, Dr. Anna Konvalinka. As a quick introduction, Dr. Konvalinka is a clinician scientist and transplant nephrologist at the University Health Network in Toronto. She is also an assistant professor at the University of Toronto and a senior scientist at Toronto General Hospital Research Institute. Her main clinical and research interests are in antibody-mediated rejection and kidney allograft fibrosis. She uses systems biology approaches and proteomics to enhance the understanding of the mechanisms, derive novel markers, and to repurpose drugs for the treatment of kidney disease. We're all very excited to have you, Dr. Konvalinka. I will hand it over to you now.
Okay, thank you very much for that kind introduction, Aisha, and thank you, CDTRP, for asking me to give this talk. I'm delighted to be here. You can hear me all right? Okay,
yes,
great.
Yes.
Wonderful. So I'm addressing you today from Toronto, which is on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. And I'm delighted today to tell you about one of the stories, or a couple of stories from my lab in Toronto. And with that, see, okay, here are my disclosures, and this is the overview of my talk. I'll first begin with a bit of the background. Kidney transplantation is the best treatment for end-stage kidney disease, which affects about 50,000 Canadians and about 800,000 Americans, and is a worldwide health problem. Unfortunately, about half of kidney allografts are lost at 10 years post-transplant, and most of it due to antibody-mediated injury. This, of course, leads to increased morbidity and mortality for the patient and high costs to the healthcare systems. So it follows that better understanding and treatments of antibody-mediated injury to prevent premature graft loss are of primary importance. It may surprise you that antibody-mediated rejection is a sex dimorphic disease, meaning that it affects men and women differently. So females are more sensitized than males through pregnancy, which makes them more prone to develop memory responses after the transplant. And interestingly, female recipients are more likely to lose their grafts compared to male recipients when the donor is a male, perhaps because of some expression of certain antigens and proteins on the Y chromosome, which only males possess. But many other kidney diseases also display sex dimorphism, most notably ischemia-reperfusion injury and diabetic kidney disease, to name a few. However, the underlying reasons for this sex dimorphism are currently poorly understood. And my lab is very interested in addressing clinically unmet needs and questions through the use of molecular methodologies to better understand underpinning molecular mechanisms and to identify potential therapeutic targets. And one such interesting methodology that emerged relatively recently is single-cell RNA sequencing. And this is really a unique opportunity to study both individual kidney tissue cells and immune cell populations. And here I've listed some efforts to do so in the kidney on a small scale. However, I was joined by Dr. Sarah Crom, who is an immunologist at my institution, and we wanted to join forces to understand kidney parenchymal and immune cell phenotypes in antibody-mediated rejection. But first, to do that, we wanted to understand what a healthy kidney looks like at a single cell level. And it may not surprise you that there's a paucity of studies of healthy kidney in literature because we never biopsy kidneys from healthy people. And there is a paucity also of the analysis of sex-based differences in kidney cells. So we had two goals. First, to define cell populations and their transcriptional or gene expression profiles in healthy kidneys. And second, to evaluate transcriptional differences between male and female single kidney cells. And I'll tell you how we went about this, but first I'll tell you how I think about the kidney at the single cell level. So kidney is a very complex organ with more than 26 different individual cell types and this unit, functional unit called the nephron. And so each one of these cell types performs individual, very specialized function and expresses a very specialized set of genes and transcriptional programs that determine that function. And if a kidney is injured through rejection, for example, or disease, then immune cells can also enter and cause additional injury, but also contribute to the overall kidney cell complement. And when we see them within glomerular capillaries like here, this is one of the key features of antibody-mediated rejection. So in a traditional paradigm, if we were to study gene expression or transcriptional profile in a kidney, which consists of many different cell types, we would take a biopsy and then obtain an average gene expression level across all these different cell types in a single biopsy. And then we would analyze that and compare it to another biopsy. In this new paradigm, what we could do is take a biopsy from a kidney and dissociate it into these individual kidney cells. And then for each of the identified cells, we would identify this particular cell transcriptional profile. And then we would put these transcriptional profiles together, and those cells that have more similar transcriptional programs would cluster typically closer together compared to the other ones. And you may have heard of this analogy to a smoothie, referring to bulk gene expression, and a fruit bowl, referring to each individual cell, such that we can tell the exact fruits that are in the bowl, how many of them there are, how they're arranged, and so on, whereas that's much harder to tell in a smoothie. And so using this single cell approach, two remarkable women in our labs did most of this work. First, Dr. Katrina McAvoy, who is a transplant nephrologist and PhD scientist, who is now a staff nephrologist in Dublin, and Julia Murphy, a PhD candidate in Sarah Crom's lab. And so Katrina and Julia decided to recruit a balanced, sex-balanced group of living healthy kidney donors. And using our protocol already developed, where we biopsy all kidneys pre-implantation on the back table, they obtained these fresh live kidney biopsies and developed a dissociation protocol to dissociate each kidney biopsy into individual kidney cells. And then these cells were either analyzed directly or subjected further to CD45 positive enrichment, which enriches for immune cells. And then in either case, the biopsies or the single cells were analyzed using 10x genomics, 3' version 3 single cell RNA sequencing. And so what we identified was over 27,000 high quality single cells. When you look at the total kidney single cell map here, you can see that PT or proximal tubular cells constituted the bulk of it. And that's not surprising since these cells are the most abundant cell types in a kidney. However, you can also appreciate that less than 1% of the cells in the healthy kidney were immune cells. So following CD45 enrichment, we were able to resolve more of these clusters of immune cells. And then we put the two maps together to obtain this map of a healthy human kidney, which contained not only the abundant kidney cells, but also some less common and more difficult to dissociate kidney cells like podocytes or parietal epithelial cells. And there, our first question was whether we could identify any differences in kidneys that come from males and those that come from females. And perhaps not surprisingly, we found most of the differences in the proximal tubular cells because we were most powered to do so with the greatest number of cells. So here I'm showing you a Verimax analysis where we clustered male and female proximal tubular cells based on their gene expression alone. And you can see that female cells cluster completely separately from male cells here on the bottom. To further confirm this, we used a different machine learning algorithm, again, looking only at transcriptional expression in these cells. And this very similarly clustered these two cell populations completely separately. So then to see which individual genes are actually driving this expression, we plotted here genes on the right that were significantly increased in female proximal tubular cells compared to those that were increased in male proximal tubular cells. And you can notice that many of these genes were actually autosomal, not on sex chromosomes. And you can see the increased expression in these 3 metallothioneins, which are important genes in antioxidant response to stress. And so we then, to validate these findings, took new, fresh proximal tubular epithelial cells for males and females, and we showed that those genes that are expressed on the Y chromosome were only expressed in male proximal tubular cells, but completely absent in female. And in contrast, 2 genes that were expressed on the X chromosome and also that escape X inactivation were significantly higher in female compared to male proximal tubular cells. And importantly, we validated the observation that metallothionein genes are indeed increased in female proximal tubular cells compared to male cells. So what we did next was we asked if there was any difference in biological processes that are dominantly mediated by male or female proximal tubular cells. And here on the bottom in orange, you can see those biological processes that are significantly enriched in male proximal tubular cells. And on the top, those that were increased in female proximal tubular cells. And so you can appreciate that male cells had an enrichment in aerobic respiration, oxidative phosphorylation, TCA cycle, nucleoside triphosphate metabolic process. So all metabolic processes that are important in energy generation. And when we saw this, we got really excited because one of my postdoctoral fellows at the time, Dr. Sergi Clotet-Freixas, who is now a PI at McMaster University, had been thinking about this particular issue for a long time. And so what we did was we then took male and female proximal tubular cells and we studied their metabolism in real time. And Sergi showed that male cells had higher basal respiration, ATP-linked respiration, reserve capacity, and maximal reserve capacity. And all of these processes result in generation of ATP and NAD, which were both indeed significantly increased in male compared to female cells, suggesting that male cells shuttle more carbons to the TCA cycle in mitochondria and generate more energy. But to really test that directly, And this is now a small part of a very large body of work that we've generated. Sergi fed male and female proximal tubular cells with heavy carbon-labeled glucose in a so-called nutrient flux analysis. And so what we're able to do in this is to follow how glucose is broken down in cells in the process of energy generation. So it can either be broken down to lactate through glycolysis, or it can enter the TCA cycle to generate more ATP. And so by feeding the cells this heavy carbon-labeled glucose, we can then at pre-specified time intervals quantify the amount of these labeled metabolites and know from their ratio how much, how quickly the cells are metabolizing glucose. So what you can see here on the x-axis is the hour of exposure to heavy labeled glucose, and on the y-axis, the quantity or the amount, relative level of the specific metabolite. And you can see that male cells had increased acetyl-CoA and a trend to increase citrate and succinate, as well as increased alpha-ketoglutarate at various time points following exposure to heavy carbon-labeled glucose, suggesting that male cells indeed shuttle more carbons to the TCA cycle than female cells when fed heavy labeled glucose. However, Sergi wanted to do this experiment with heavy labeled glutamine, since glutamine, again, is an important amino acid, which can be converted to glutamate and then alpha-ketoglutarate, which subsequently can enter the TCA cycle and also participate in the generation of ATP. So this time, Sergi fed heavy carbon-labeled glutamine to proximal tubular male and female cells, and he found that male cells had significantly higher alpha-ketoglutarate, malate, and citrate, particularly at half an hour and 1 hour following exposure to heavy labeled glutamine, further confirming the original observation with heavy carbon labeled glucose and suggesting that perhaps male and female proximal tubular cells shuttle their carbons to the TCA cycle differently and generate energy in different ways. So I will switch gears a bit and tell you a little bit about what we found with respect to the immune cells in the kidney. So when Julie and Katrina reclustered the immune cell clusters, they were able to generate these 3 dominant cell clusters, and we were most interested in this one mononuclear phagocyte cluster called MP1. And these were this was a cluster of mononuclear phagocytes in the kidney that expressed markers of alternative activation and macrophage markers. So we were interested in this cluster because when we integrated our data set with other external data sets, 3 other external data sets that also analyze kidney mononuclear phagocytes, we were able to generate this map here representing all the different mononuclear phagocytes data sets that were identified in these 4 studies. The green arrow points to the cluster of these alternatively activated macrophages MP1 that we identified. And so when we look at the feature map shown here, you can see that this cluster was the main cluster in our living donor data set, whereas it was almost completely absent from the other 3 data sets in literature, suggesting that we were identifying a unique population of kidney macrophages that other groups hadn't been able to find, perhaps because of their susceptibility to dissociation, or perhaps because disease conditions tend to deplete them. This is currently an open question. But we also decided to look at T and NK cells in the kidney because of their important role in antibody-mediated rejection. And what we found was first that kidneys exhibited higher proportion of NK and NKT cells compared to peripheral blood mononuclear cells, and we verified this using flow cytometry. And interestingly, kidneys also exhibited a higher proportion of CD8 positive T cells compared to peripheral blood. And both these CD8 positive and CD4 positive T cells exhibited a memory phenotype as evidenced by the expression of this protein CD45 rho. And this was in contrast to peripheral blood, where majority of T cells are naive. And when we specifically looked at this population of memory CD4 positive T cells, we noted that their transcriptional profile suggested a Th1/17 phenotype. And so to conclude this first part, I showed you that our single-cell RNA dataset may be particularly useful as a baseline for studies of acute and chronic kidney diseases, since this is sort of the biggest, probably the biggest data set of normal healthy kidney single cells. And we show for the first time that proximal tubular cells from males exhibit distinct transcriptional profiles and higher rates of oxidative phosphorylation compared to female cells. And that in contrast, proximal tubular cells from females display increased expression of antioxidant metallothionein genes. We identified a unique macrophage cell population in the living donor kidneys. And we also demonstrated that living donor kidneys contained higher proportion of CD8 positive T cells, NK cells, and memory T cells compared to peripheral blood mononuclear cells. But of course, we're interested in what happens to cell infiltrates and distinct compartments in kidney antibody-mediated rejection. And so when I think about antibody-mediated rejection, I think about mainly events that take place in the microvascular capillaries, so glomerular and peritubular capillary network. And there within these networks, donor endothelial cells sit on the basement membrane and constitutively express a human leukocyte antigen or HLA class 1. And quite often under conditions of activation and injury can express HLA class 2. And if these are different enough between the donor and recipient, the recipient can develop donor-specific antibodies or DSA against these class 1 and class 2 antigens. And it's generally accepted that these DSA can injure endothelial cells either directly through complement activation or through antibody-dependent cell-mediated cytotoxicity, whereby the Fc domain of the antibody ligates Fc receptors on myeloid or NK cells, resulting in production of pro-inflammatory cytokines. And lastly, a particularly ominous form of chronic antibody-mediated rejection is called transplant glomerulopathy, whereby the basement membranes are significantly remodeled. This condition carries poor prognosis and is incompletely understood. However, a conundrum is that based on different studies, roughly 30 to 60% of individuals with donor-specific antibodies do not experience rejection. And this is one of those questions that we have set out to answer using a variety of different methods. And I'll first show you how we have been using some protein-based profiling of antibody-mediated rejection. First, I sought to identify kidney allograft recipients at University Health Network who were followed here between 2007 and 2022. And that had information about donor-specific antibodies and that had a for-cause kidney biopsy. We don't do protocol biopsies, so these are the only biopsies that we do. We identified over 2,500 patients and excluded those who with documented DSA but no biopsy, or those with undocumented or uncertain DSA. So we were left with 240 patients with 260 biopsies that had DSA and a concomitant kidney biopsy. Of these, 60 patients with 61 biopsies had no rejection, and 181 patients with 199 biopsies had both DSA and antibody-mediated rejection. And to be sure, these patients had either acute antibody-mediated rejection, chronic active antibody-mediated rejection, or mixed antibody and cellular rejection. So we first asked if there were any clues in the biopsy that could help us understand why patients with DSA have such distinct outcomes. And so the first thing that I will show you is preliminary data, new preliminary data that we have from a laser capture and microdissection of these biopsies. This is a method that was first pioneered by Sergi Clotet Freixas while he was a postdoc, and we published this study in JASN in 2020. And currently, Dr. Kieran Magnon, who is an outstanding postdoctoral fellow in my lab, is working on using this same method to answer the questions. So with the help of our renal pathologist, Dr. Rohan John, we took these archived biopsies from 24 patients with antibody-mediated rejection and DSA, and 18 patients without rejection despite DSA. We took 18 micron sections from formalin fixed paraffin-embedded kidney biopsies. And this is what the biopsy would look like under the microscope. And using a laser, we would specifically cut out either glomeruli or the tubular interstitium is shown here. And we'd normalize that by surface area on a per biopsy basis. So we then separately digested proteins into peptides from glomeruli and separately tubules, and then analyzed these peptides using LC-MS on a Q-exactive mass spectrometer in order to identify all proteins present and to quantify those proteins that were significantly differently expressed between patients with antibody-mediated rejection and those with no rejection. And so just to show you some clinical characteristics of these two groups of patients, most of these patients were male in both groups. Their age and biopsy was similar, although patients with ABMR were slightly younger. And importantly, patients with antibody-mediated rejection tended to be biopsied later after transplant. We also examined clinical and histopathological characteristics. And as you can see, patients with no rejection had a roughly equal proportion of class 1 and class 2 DSA. And in contrast, patients with antibody-mediated rejection had predominantly class 2 DSA. When we examine microvascular inflammation, such as glomerulitis and peritubular capillaritis, both of these were present, of course, only in patients with antibody-mediated rejection. None of these patients in this first group had chronic glomerulopathy or transplant glomerulopathy, and there was a distribution of patients either without or with C4d complement deposition. So we were able to quantify over 1,400 proteins in the glomeruli and over 2,300 in the tubular interstitium. We used a rigorous cutoff to eliminate all those proteins that were detected in less than 50% of all samples. And we imputed missing values using normal distribution and then performed a t-test followed by Benjamini-Hochberg adjustment for multiple hypothesis testing. And we're able to finally identify 13 significantly differentially expressed glomerular and 8 tubular interstitial proteins. And so I'm showing you these proteins here, glomerular ones on top volcano plot and the tubular interstitial ones on the bottom. All those proteins shown on the right side are significantly increased in antibody-mediated rejection, and those on the left are significantly increased in no rejection. I'll draw your attention to this protein CD74, which was significantly increased in both glomeruli and tubular interstitium of both biopsies. So this we found to be an interesting protein because CD74 actually complexes with MHC or HLA class 2 in the Golgi apparatus and prevents MHC class 2 complex from being refolded prematurely before the peptide is integrated. So it enables adequate integration of the peptide followed by presentation of this peptide to CD4 positive T cells on the surface of the cell. And CD74 is also a receptor for ligands such as macrophage inhibitory factor and a couple of other factors. So previous preclinical studies have shown that inhibition of MIF and CD74 interaction ameliorates autoimmune diseases like experimental lupus, crescentic glomerulonephritis, or IgA nephropathy. And an old study in 2003 showed that when MIF knockout mouse kidney allografts that are lacking MYF in all of its cells and tissues were transplanted into a fully MHC mismatched recipients. These mice were not protected from rejection. Nonetheless, there is some new data that was just presented at the ASN by Jamil Aziz's group, which shows that CD74 complete knockout mice, when they had an MHC mismatch cardiac allograft transplant, they lived indefinitely. And preliminary results indicate that this may be mediated by T regulatory cells, intriguingly. So we went back to our single-cell RNA sequencing study in healthy donors and said, well, where is CD74 expressed? And we found that CD74 is mainly expressed in these mononuclear phagocytes, particularly the cluster 1 that I showed you and another cluster 3, but is also expressed in B cells and endothelial cells. And if you're interested in plugging your favorite molecule, you can go and look up this a web platform, which is free and open access, and play with it. We then wanted to see where MYF was expressed, which is a ligand for CD74. And in contrast to CD74, MYF was expressed mainly in proximal tubular cell and other epithelial cells in the kidney, suggesting that perhaps proximal tubular and other cells interact directly with mononuclear phagocytes in the kidney. And bringing into question whether spatial transcriptional and proteomic methods could be useful to delineate these questions. And so what we have been working on was to develop methods using so-called imaging mass cytometry. And this is a project of Maya Allen, who is a master's degree student in my lab and soon to be a PhD student. And what we can do with this method is tag up to 40 different antibodies with different heavy metals, then combine them and add them to the slide with a patient kidney biopsy, then shoot the laser. The laser then releases these metal tags. And because it only shoots in a specific location, we can get information about the metal quantity as well as its indexed location. And in this way, we can spatially annotate even single cells. So what Maya did was Maya first examined the biopsy with no rejection despite DSA. And what you can see here is the same region of interest shown. In blue are always nuclei. In red is CD74. In green here on the left is nestin, which is a marker of podocytes in the glomeruli. In the middle, green is CD31, which is a marker of endothelial cells. And on the right is CD45, which is a marker of immune cells. And if we zoom in, you can see that CD74 is not expressed by podocytes, as you see no yellow here, no co-expression with nestin. It is expressed with some endothelial cells in the glomerulus and an occasional CD45 positive immune cell expresses CD74, although you don't see that many immune cells in a no rejection case. Maya then decided to examine an antibody-mediated rejection case. And here, the staining is exactly the same with the exception of the right region of interest, which as a green has CD68 marker, and that's a marker of macrophages or these mononuclear phagocytes. And when we zoom in, we can see again that nestin doesn't really express, nestin labeled podocytes do not really express CD74. CD74 is expressed by a few CD31 positive cells, but it's mainly expressed by these intraglomerular macrophages. And then in another part of this biopsy, we noted, interestingly, that there was quite concentrated CD74 staining. The other two stains are identical, but the stain on the right shows in green CD20, which is a marker of B cells. And so when we zoom in, we can see that in this particular area of the biopsy, Podocytes definitely do not express CD74. There is no CD74 in endothelial cells. However, it's mostly B cells that express CD74. And this illustrates kind of the importance of spatial information, because if we weren't looking at this from a spatial context, we might have completely missed this observation. However, antibody-mediated rejection is not a uniform disease. I told you in the beginning that out of all of these 180 or so patients who had antibody-mediated rejection, some of them had acute, some had chronic active, and some had mixed antibody and cellular rejection. So we're currently in the process of expanding data analysis, but in doing so, we decided to parse out proteomic expression in these different subsets of antibody-mediated rejection. And so what I'm showing you here is a heat map where you can see patients clustered based on their diagnosis with acute antibody-mediated rejection, chronic active, mixed rejection, or no rejection. And here's a summary of their expression with some proteins and their signature pathways. And if we zoom into this area, what you can see is clear differences in patterns of expression. So acute antibody-mediated rejection, for example, has the highest expression of this cytokine signaling and immune system signature. Whereas, interestingly, chronic active antibody-mediated rejection has in red increase in proteins that are correspond to extracellular matrix organization and complement. And when we looked at the tubular interstitium, where you can see the exact same patients, except now we're analyzing proteins in their tubular interstitium. And if we zoom in, you can see that these proteins in the first box are all related to interferon signaling, which interestingly was the highest in mixed and cellular rejection. The complement system is again highest in chronic active antibody-mediated rejection, which is an interesting observation that we will be following up on, particularly the observation that many of the terminal complement components are found here. And lastly, metabolic proteins were the highest or probably most preserved in patients with no rejection, suggesting that the importance of metabolism in the tubular interstitium, particularly as pertains to proximal tubular cells. So to conclude, our preliminary glomerular and tubular interstitial proteome of biopsies with antibody-mediated rejection suggests expression of CD74 protein. This particular protein appears to be expressed by CD68 positive macrophages, but also by B and endothelial cells. Our further studies will focus on the integration of molecular data sets from this cohort and will turn to probing potential mechanisms in vitro and then in vivo. Just to show you a bit of a paradigm that my lab is thinking along and working towards a potential precision approach. So you can see this cohort of patients who are DSA positive and had kidney allograft biopsy. I've already shown you some of the findings that we are beginning to tease out with respect to the proteome that reflects antibody-mediated injury compared to the proteome that reflects accommodation in those patients who are DSA positive but have no evidence of rejection. We're also currently in the process of analyzing data from a subset of these patients who had single-cell RNA sequencing of their fresh kidney biopsies. And this is again work with Dr. Krohn's lab. We will integrate these two data sets to perhaps delineate which cells are expressing the most interesting proteins, and also to potentially infer some underlying causal mechanisms that can be tested in vitro. And with the help of imaging mass cytometry, we're hoping to define single-cell spatial proteome. And lastly, with the help of our collaborators from US, we're able to analyze donor-specific antibodies from a concomitant blood sample compared at the time, collected at the time of biopsy from these patients, where we're hoping to define potential biochemical properties of these antibodies beyond their specificity or MFI. And so with that, I will recognize the people who do the work. Work was mainly done by Dr. Katrina McAvoy, Dr. Scholey and Dr, Dr. Kieran Mannion, and Maya Allen. And lastly, I will recognize my collaborators, Shengwei Su, our tireless biobank manager, who makes sure that we can indeed move towards precision medicine by studying patient-specific biological samples, patients and their families for generously donating both their sample and their clinical information and the sources of funding. And with that, I will end, and I would be happy to answer any questions.
Thank you for that excellent talk. I'm going to open the floor for any questions. Please go ahead. First hand up.
Hi, Anna. Great talk. Thank you. I just wanted to take a step back at how you previously defined differences in the metabolic processes between male and female tubular cells. So have you or will you investigate whether these metabolic differences are linked to the female hormone cycle? Only because I was thinking that some of the research suggests that hormone imbalances in women do correlate to dysregulated metabolic pathways.
Mm-hmm. Yeah, very interesting question, Dimitra. Thank you. I will answer a little bit differently. I mean, I think in general, we have not studied whether hormonal cycles in females affect this at all. We're not really positioned to study that because we're getting these cells from Lonza. We're purchasing them from Lonza. They're obviously primary kidney cells, but they're isolated from presumed healthy individuals. So we have their age and sex, but we have no idea, about their cell cycle and so on. However, this big manuscript that was just accepted in Science Translational Medicine, we have actually tried to delineate the mechanism that underpins this observation. And some, it's not entirely clear, but some of it does relate to sex hormones. So for example, this kind of shuttling of the carbons more to the TCA cycle and predominantly mitochondrial metabolism in male cells is indeed attributed likely to male sex hormones. However, we also identified this histone demethylase KDM6A, which is X-linked protein that escapes axin activation, and that very interestingly has a different role in male and female cells and in the context of hyperglycemia or kidney injury. So, I think it's more than just one thing, but I think this particular observation pertaining to metabolism may be more mediated by sex hormones than sex chromosomes.
Okay, that's very interesting. Yeah, I know the relationship between the metabolic cycle and hormones isn't fully understood, especially in certain disorders as well in women. But I know that there's been findings like increased calcium could have impacts on the citric acid cycle, and that could be linked to hormones. So I was just curious if you were looking at that information. So anyways, thank you.
Thank you, Dimitra. I'm going to hand it over to Andrew.
Thanks for the stupendous presentation. It's mind-stimulating, and the field is really top-notch and well needed in terms of precision medicine for the vulnerable patients of transplantation. Two questions. First one, what are your speculations about extending your current study to include aged population, for example, aged females, given that the sex hormones levels will be different from the adult population, and given that they are underrepresented in research. The second question would be tracking the senescence in terms of the mitochondrial level, like thinking about mitochondrial senescence in terms of on a single cell level, both in males and females. What are your speculations on both? And thank you very much.
Okay, two very interesting questions. So first, yes, you're absolutely right that some female protection is lost after menopause, clearly implicating sex hormones, and that's the case in diabetes, for example, diabetic kidney disease, and that's also the case in ischemia or perfusion injury, where in animal models this dimorphism was attributed to sex hormones. We haven't gone there as yet, but Sergey, who was the postdoc who really drove this project, before he came to the lab, I'd never even thought about or heard of these, and then he kind of just pushed me there. And then it became more and more interesting. So, so I think in the end, then we were just wanted to satisfy our curiosity. So these are some of the questions that he's pursuing in the context of, how, to what degree age, for example, and sex hormones can contribute to some of these observations. And one thing I will say is that there is a very interesting mouse model that we leveraged in this paper with the help of this professor emeritus Arnold from California, from UCLA, who basically can dissect, it's called the 4 core genotypes mouse model. So he can dissect the effect of sex chromosome from gonadal sex. So basically in that way, kind of separating the effect of sex chromosomes and sex hormones. So I think that's kind of the best way that I would pursue this, because aging also, as you mentioned, senescence, aging also adds another layer of complexity. So with respect to senescence, I think that's a very interesting question and an important topic. And of course, there are several groups that are studying senescence following kidney transplantation. And it's been recognized that kidney transplants tend to be more senescent and age faster following transplantation for reasons that are not completely understood. So I do think that this is a fertile ground. We're looking at to see, for example, if some of these differentially expressed proteins in different forms of rejection or had been implicated in senescence, for example, or if time post-transplant, which I showed you is an important, and it is always an important factor in these molecular analysis. We're trying to see if at the protein or gene expression level, these senescent programs, there is evidence that these senescent programs may be activated, so that we would study them further, but I don't have an answer as yet.
Thank you very much. Yeah, I think replicative senescence and inflammation would be very interesting in your model. I would be curious to see, would patients receiving hormone replacement therapy have a different biopsy phenotypes, for example, because they are having an external source of treatment in terms of HRT or hormone replacement therapy. But as you mentioned, it's a very interesting area in research and the possibilities are endless. So thank you very much.
Katie.
Great questions, Andrew. I'm going to open it up to Katie.
Hi, Anna. Thank you for that talk. I really, I thought your images were so beautiful, and your smoothie fruit bowl analogy, very, very, very good. It's a great way to show what that looks like to someone who doesn't have necessarily the basic science knowledge. My question for you is about the unique population of living donor kidney-specific macrophages that you group identified. What are your next steps to explore this cell population?
Yeah, that's great. I mean, I'll admit to you that the analogy is not, I did not coin it. It's been used and reused in different presentations, but I also thought it was kind of a clever way of showing.
I like it.
But with respect to macrophages, yes, we were quite intrigued, and I often get asked something along those lines, like, why would there be this unique population? And, I think, there are several different possibilities as to why we were the ones to identify them. It's interesting that Slava Eppelman, who is at my institution, also a clinician scientist and cardiologist who is studying kind of macrophages in the context of the heart, was actually able to identify similar population in mice. So, they're clearly, conserved across at least humans and mice. I don't know if they're more susceptible, because in general, these particular cell types are quite susceptible to dissociation. So basically they kind of die, or, they don't tend to tolerate dissociation very well. So it's possible because this was a very controlled setting and kind of very healthy kidney tissue that's why they survived in our hands. It's also possible that they disappear, or they're destroyed in the context of disease, and that other macrophages take over. For example, now we have preliminary data actually from these patients with antibody-mediated rejection and those with no rejection. And this cluster is still visible there, but a more dominant cluster becomes this cluster exhibiting markers of inflamed macrophages, perhaps not surprisingly. So in that first paper where we published on these macrophages, we tried to isolate them from the biopsies directly, and we were successful in small numbers. So we were successful at showing, using flow cytometry, confirming that indeed there is a population of these macrophages that are higher here and that express these specific alternative markers of alternative activation. But I don't know as yet how feasible it is to culture them outside. Something that we're kind of interested in trying in the context of several other related studies. So I may have an answer in a few years, but I don't know if there are going to be enough, because I showed you in a healthy kidney there are less than 1%. So I think it would have to be more of them in the kidney for us to be successful at isolating them and even culturing them. But
thank you.
Yeah,
that was going to be my follow-up question was, do you think that you'll be able to culture them? I was just typing to Dimitri, I was like, can you culture those? And you spoke to it. So thank you so much, Anna.
Thank you, Katie.
That was a great answer. And I'm glad Katie asked that, because I had somewhat of a similar question. But with the remaining few minutes, I have a question from the chat from Erica. The question is, are you aware if the sex dimorphism you reported are also present in children or only adults?
Oh, great question. No, I'm not aware about children. So that's another interesting question, kind of before puberty takes place. So I don't know. However, what I will say is that, in this paper, we were kind of pushed by the reviewers to look at, because, and the first population we wanted to look at was patients with diabetes mellitus, because diabetes mellitus clearly affects metabolism of all cells, including kidney cells, and that metabolism is implicated in disease progression. So we looked at 3 different populations across the spectrum of diabetic kidney disease, and one of these 3 populations was a population of youth with type 2 diabetes. So it was a kind of a unique population of youth, indigenous youth from Manitoba. So we did have that one population, and I think, of course, we can't do these kinds of studies that we did in cells and that we could do in mice, but we could measure their metabolome in blood And so we did that across these 3 different cohorts. And we found that many of the same observations as in cells still hold. So, males tend to have higher DCA cycle metabolites in blood. And actually in bigger and more advanced, more aged patient cohorts, some of these DCA cycle metabolites actually in diabetic and established diabetic kidney disease actually correlated with mortality. So it's quite interesting that we were able to see that in blood, suggesting that it's probably not just proximal tubular cells that exhibit this phenomenon. It may be multiple other cell types in the body.
Thank you.
Very cool. So I see that time is just about up, and it's great timing, Dr. Konvalinka. Thank you for the questions and comments throughout today's presentations, everybody. And Dr. Konvalinka, on behalf of CDTRP, thank you again for presenting. If anyone has additional questions, comments, or suggested topics and speakers, please contact Erica Croft at ekcroft@cdtrp.ca. I want to mention that all participants will receive an email asking them to fill out a survey, and we would appreciate it if you could take just 3 minutes to complete it. With that, thank you everyone, and enjoy the rest of your day.
Thank you. Bye.